how many people in the room could start to rattle off the names of strategies for mitigating biological threats? And speaking as somebody who is a practitioner both of the biotechnology, but also in the circles of biosecurity to a degree, there's just a gap there. It's not that there aren't good ideas, but those ideas aren't being turned over and debated and worked through with the vigor and rigor that I would wish to see, let alone being coupled to strategic investments in public health, in science and technology development. Again, it's not that such things aren't happening, but they're happening on a very idiosyncratic and ad hoc basis. And to the extent that biology is central to many of the things we care about, starting with ourselves, but certainly the environment, and to the extent that our civilization is going through a potential transition.
So just to back up a second, because there may be some that are a little bit bewildered and you're not quite up to speed on some of the terms we've used. So let me just say that what we're looking at now with CRISPR is, and that's an acronym and it doesn't matter what it stands for, but what it turns out it's a very ancient mechanism, billions of years old, that microbes have used, particularly bacteria, to expel unwanted entry of genetic material brought in by phage or viruses that invaded the bacteria. So you can think of it as a roughly 3 billion year old immune system. It's only very recently that our species, Homo sapien, discovered this ancient thing that's been out there forever and has been the way that bacteria maintain their genetic integrity against the constant onslaught of phage in their environments. And probably the way they plucked useful things from the phage when they needed them, such as the capacity to override antibiotics. Now, the problem is we discovered this. It turns out to be so easy. Wow, boom, cheap. And then discovered is another layer to it, which is gene drive, which allows you to essentially tell a genetic segment to just keep on running and to run in the next generation and the next generation and the next generation so that it becomes a permanent feature on the landscape. Now, just last week, I was web in on a briefing at the NIH where the heads of 2 institutes were briefing the head of the NIH about Zika. It was a terrifying meeting. But at one point, Francis Collins asked Tony Fauci, the head of the National Institute of Allergy and Infectious Diseases, well, what about genetically modified mosquitoes? Can't we use CRISPR to take care of this problem? And then we won't have mosquitoes carrying dengue, chikungunya, yellow fever, and Zika. And Tony very wisely said, well, there's 2 problems with that idea, even though the stock market, by the way, really likes it and is spending a lot of money in this. One, these mosquitoes only travel in their lifetime about 200 yards. So if you're going to release a single generation CRISPR, it's going to have to be released every 200 yards, which is a pretty tough way to control mosquitoes. And then if you're going to say, well, no, we want it to go on forever and make a gene drive technology, you better know what you're doing. And you better have a really good idea how you're affecting evolution out there if you deliberately and permanently alter Aedes aegypti mosquitoes. And Tony's advice was, let's not do it. So that gets to what Drew said. Where's that wisdom over time that builds a series of baselines about the yes and the no? Where are the lines? How do we decide? What's the process? Where do we go? Many elements of what we're in right now as a debate, a brand new debate in its infancy, overlaps with starting with the Asilomar 1975 meeting when scientists themselves said, whoa, genetic engineering, if we alter viruses, what might happen? Let's all come together and make a rule book. That went on for some time in some permutations. Then in early 2000s, we had dual use research of concern, DIRC, arose because of deliberate modifications of flu viruses that were going on to make supervirulent viruses and to determine how likely they were to infect human cells. This raised a mountain of concerns among bioterrorism and national security experts and brought all these issues to the fore again. Then the similar pattern arose just a few years later with so-called gain-of-function research, or GOF, the idea that you would deliberately insert a genetic capacity into a microorganism in order to see if that made it more virulent or that made it more transmissible across a broader range of species. Once again, all the red flags went up. Everybody freaked out. The same debate over and over again. And now here we are in the next stage of the revolution. And I think Drew's absolutely right. We don't have a learning curve. We have a very tiny cluster of people in most of the key high-tech countries of the world who, when we have meetings, we see familiar faces. Oh, right, I saw you 10 years ago at the Dirk meeting, you know, here we are again. And yet we don't seem, as Drew's metaphor is perfect, we don't see, as we did with nuclear, both fusion and fission research, a permanent raising of the bar of the discussion so that each layer is a learning curve over the prior and the sort of assumptions among nations improve. And I would just add that, as a final note, that, you know, everything that we think are the values and the norms that we would aspire to in the United States on these issues are quite different in other countries.
Sure.
And we don't even have to talk about Russia, China, you know, Iran, Pakistan, what have you. Let's just look at the UK. Their Human Subjects Committee decided it was fine to make alterations in gametes with the intention of improving in vitro fertilization. So something that we think is kind of taboo here, and we haven't even had an effort to consider it formally, is already okay in the UK.
Julie, since
We'll
Lori's last remark is about medicine, medicine has an old view, do no harm. Are you worried about the ethical implications of gene editing in terms of the progress of medicine?
Well, absolutely, particularly in the germline example that Lori just gave. I think we have to be extremely cautious in that front. But, you know, and I totally agree that we need a system of risk assessment and of the rule book, so to speak, that applies globally, although I also believe that's going to be a very hard energy to harness. But it's also true that we've been here and done this before, not just going back to the gain of function opportunity. It was part of that conversation at CDC, since we were part of the science behind all of that. But, you know, go back to even, you know, smallpox vaccine or you know, the early days of vaccinology, where the same debates occurred between the technology of advancing the opportunity to prevent infectious disease versus the safety concerns and the academe. So these are ongoing tensions that exist between scientific advances and concerns for unknown risks. I can't really think of an example where the science has stopped because of the safety concerns, but I do think that the boundary conditions are important to face up to, and in this case, the stakes are particularly high.
Do you think we've made any mistakes so far with respect to the potential medical applications?
Well, what we might not understand are the mistakes that we've actually made. And, you know, the biome is one example where I don't think we really fully understand what our manipulation, even our antimicrobial use, for example, or anything that we put into our bodies, what's that really doing to our internal ecosystems, let alone the environmental ecosystem. And if you take an ecological approach and really think about emerging infectious diseases and their zoonotic origins about 80% of the time, we really probably have underestimated the perturbations that we've caused in the system. So I don't have a startling example to give you, but I have a strong sense that we probably don't know what we don't know.
Oh, that's a good way of putting it. Can I